Welcome to the deep dive. When we usually talk about a medical diagnosis or like a physical therapy, there's this ingrained expectation of precision.
Yeah, it feels almost like engineering, right?
Exactly. Like you break your arm, you get an x-ray, and there's that jagged white line right on the film.
Mm-hmm.
And the doctor points to it and says, look, right there, it's incredibly clear cut. But the moment you step into the world of gene editing or neuromodulation, it's like that x-ray machine suddenly breaks down.
Oh, it really does. Things get incredibly messy, blurry, and honestly, highly invasive.
Yeah, totally invasive.
Because for decades, we've had to rely on these very blunt instruments. We flood the entire body with chemical drugs.
Which have terrible off-target side effects, right?
Exactly. Or we resort to literally physically drilling into the skull to insert fiber optic cables for light-based therapies. It's always a game of trade-offs.
Right, but what if I told you that you could act as your own genetic DJ?
A genetic DJ, I like that.
Yeah. Imagine having a completely invisible magnetic remote control that lets you turn your DNA on and off at will.
Just at the push of a button?
Exactly. No pills to swallow, no invasive surgeries, no system-wide chemical side effects. I mean, it sounds like pure science fiction, doesn't it?
It does sound like science fiction, but that is the hardcore reality we're unpacking today.
We love to make the abstract concrete on this show, and this technology literally turns an invisible magnetic field into a molecular scalpel.
It really is a scalpel.
So today's mission is to completely decode a groundbreaking 2026 paper from the journal Cell. We're going to strip down something called the I gene switch.
Which stands for electromagnetic field-inducible system.
Right, the I switch. We're going to figure out the exact microscopic machinery that makes this invisible remote control work, and then we're going to walk you, the listener, through 3 absolute jaw-dropping applications the researchers pulled off.
Reversing cellular aging, completely rethinking how we model Alzheimer's disease, and curing severe depression.
It's a huge lineup, so let's just get right into it. How on earth can an invisible magnetic field talk directly to human DNA?
Well, to really grasp the magnitude of this, we have to look at why previous attempts failed.
Okay.
If you want to turn a specific gene on or off inside a living animal, you traditionally use drugs or light.
Right, the blunt instruments we mentioned.
Yeah. Drugs go everywhere in the body. They don't just hit
Yeah.
the target. I mean, they hit the liver, the kidneys, the heart.
So you get all the toxicity.
Exactly.
Oh.
Now, light, which is used in a brilliant field called optogenetics, is highly precise, but it has a massive physical limitation.
Can't go through walls.
Right. Light cannot penetrate deep into solid tissue. So if you want to control a gene deep inside a mouse's brain, you literally have to drill a hole and insert a physical fiber optic cable.
Which completely ruins the whole idea of a non-invasive treatment.
It really does. So the researchers in this Cell paper started looking for a physical force that naturally passes right through bone and deep tissue without causing damage.
And they landed on magnets.
Yeah, they turned to electromagnetic fields, or EMFs. But, they didn't just blast the mice with random magnets.
Yes, of course not.
They found that a very specific parameter worked absolute magic.
Mm.
We're talking about a frequency of 60 hertz at an intensity of exactly 2.0 millitesla.
Okay, I think a good way to picture this for everyone listening is to imagine you are sitting in an old car, right, turning the dial on an analog radio.
Oh, that's a great analogy.
Thanks. Because ordinary magnetic fields, the kind you get from your fridge magnets or standard electronics, are just static. It's just background noise.
Yeah, random noise.
But this specific parameter, 60 hertz at 2.0 millitesla, is like finding that perfect crystal clear radio station. The paper actually describes it as a direct current pulsed sine square wave.
Which sounds very technical, but...
But if you visualize that, it's essentially a rhythm. It's a rapid burst of 20 microscopic pulses, followed by a brief rest, and that entire sequence repeats every 16.67 milliseconds.
That radio analogy is spot on, because when the researchers tuned the EMF to that exact station and exposed the mice to it, they ran a massive single-cell RNA sequencing analysis on the brain tissue.
So they basically looked at everything.
Right. They looked at tens of thousands of genes to see what was happening. And out of all that genetic noise, almost exclusively, one single gene woke up and was significantly upregulated.
Just one gene out of thousands.
Just one. It was incredibly selective.
Yeah.
And that gene is called LGR4.
Wow. Okay, so they found the one gene that was listening to the radio station.
Exactly. So they zeroed in on LGR4. They went into its promoter region, which is the part of the DNA that acts like the ignition switch for the gene, and they extracted a tiny snippet.
How tiny?
just 450 base pairs long.
Yeah.
Very small. They named this sequence the I element.
The I element. Got it.
Then they took this I element and attached it to a completely different neutral reporter gene just to test it out. And it behaved like the perfect biological light switch.
Meaning when the EMF is on, it's on.
Exactly. When they turned the EMF on, the gene turned on. When they turned the EMF off, the gene shut down completely within 24 hours.
That's amazing. But didn't they optimize it even further? I remember reading about a faster version.
Oh, they did. They built a second-generation version called the Psi system. They added a specific enhancer and managed to get that activation time down to just 2 hours.
2 hours, that is just incredible. So to pause for a second, we found a magnetic frequency that easily penetrates deep tissue, and it acts as an on and off switch for a specific snippet of DNA called the I element.
So that is the foundation of it all, yeah.
It really makes you wonder, right? I mean, to you listening, if you could safely turn on just one biological trait in your own body using a magnetic switch, what would you choose?
Right. Well, there are so many possibilities.
Right. But before we get to the actual applications they tested, I have to stop you here. I am struggling with a massive logical gap.
Okay, let's hear it.
We are talking about magnetic fields, which are pure physical energy, but DNA, proteins, cells, that is a biochemical system.
It is.
So how exactly does physical energy cross the bridge and translate into biochemistry? The physical wave has to turn into a chemical signal somehow. There has to be a translator.
That is the exact right question to ask. And it's where the researchers did some of their most, well, elegant work. They knew there was a missing link.
Okay.
So they ran a colossal CRISPR-Cas9 genome-wide screen. Imagine having a massive soundboard with 20,000 switches representing every single gene in the cell.
Wow, 20,000.
Yeah, and they systematically knocked out those genes one by one and exposed the cells to the magnetic field. They were basically looking for the one broken switch that made the cell go deaf to the EMF.
Talk about a microscopic needle in a haystack.
Truly. But they found it. They identified a gene that codes for a protein called Cy5b.
Cy5b?
Right. Now, Cy5b is a membrane-bound electron carrier. It sits right on the outer skin of the cell. And this protein acts as the actual physical antenna for the electromagnetic field.
Okay, so C5b is the antenna. It catches the 60 hertz radio station. What happens next? How does the antenna tell the DNA to actually turn on?
Well, when Cy5b senses that specific magnetic field, its oxidation state changes.
So it physically changes shape or charge.
Exactly, a physical shift. And that physical change acts as a trigger for another structure sitting right next to it on the cell membrane, called CACNA1.
CACNA1, okay, what is that?
This is what we call an L-type voltage-gated calcium channel. You can think of it as a heavy locked gate on the castle wall. When the antenna gives the signal, the CACNA1 gate swings open, and calcium ions from outside the cell suddenly rush inside.
Okay, this is where it gets absolutely brilliant, and I want to offer another visual for you listening. Think of that calcium rushing into the cell as a sound.
A sound, I like that.
Right, because if you heat up a cell or you hit it with a heavy chemical drug, the calcium channels get forced open and the calcium just floods in all at once. It would sound like a continuous blaring alarm, just a solid, loud beep.
Right, a total system flood.
But under this specific magnetic field, the researchers used real-time fluorescent imaging and saw something totally different. The calcium wasn't flooding, it was pulsing.
It was a rhythmic calcium oscillation.
Exactly. It sounded like beep, 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 beep.
Beep, beep, beep.
Yes. It is quite literally a biological Morse code. And this is the key to why this entire system is so incredibly safe.
Because it's a specific password.
Exactly. In biology, we use the term bio-orthogonal. That means a process that doesn't interact or interfere with the native natural biology of the organism.
So it's basically invisible to the rest of the cell.
Right. The reason the magnetic field doesn't cause chaos in the body is because the cell's normal machinery completely ignores that specific Morse code. A massive flood of calcium might trigger a cell to panic or even die. But that specific beep, beep, beep rhythm, it only wakes up one highly specialized transcription factor inside the cell.
What's that called?
SP7.
So SP7 is the decoder ring. It's the only thing in the cell that actually understands the Morse code.
Precisely. So SP7 hears the rhythm, gets activated, travels down to the DNA, binds directly to that I element we talked about earlier, and turns the gene on. That is just a flawless chain of events. So, okay, let's take a quick moment to lock these concepts in with a fast 5-minute recap for you listening, because these details are the foundation for everything that comes next.
Good idea.
Bullet 1. This specific EMF penetrates deep tissue perfectly, bypassing the physical limits of light and the chemical side effects of drugs.
Exactly.
Bullet 2. A protein called CYP5B acts as the biological antenna on the surface of your cells.
The receiver.
Right. And bullet 3, that antenna opens a gate that taps out a rhythmic Morse code using calcium, which is the secret password to turn on the gene.
A very precise sequence.
It really flips your perspective on biology. Just knowing that your microscopic cells possess the capability to decode specific physical rhythms, I mean, it's mind-blowing.
It completely bridges physics and biology.
Right. So now that we have this perfect remote-controlled switch, what happens when we wire it up to something powerful? What happens when we attach this switch to the genes that actually control aging?
Oh, this brings us to the first major application in the paper. In vivo partial reprogramming.
Reversing time.
Basically, yeah. The researchers decided to hook the eye switch up to something called the OSK factors. That stands for octone 4, SOX2, and KLEVA4.
OSK.
Right. These are essentially the cellular factory reset buttons.
Yeah.
When you turn them on, they wipe away the epigenetic marks of aging and revert an old cell back into a youthful state.
But there is a massive catch here, isn't there? I mean, biology never gives you a free lunch. If you just press and hold that factory reset button, things go terribly wrong.
Disastrously wrong, yeah. If you leave the OSK genes turned on for too long, the cells completely forget their identity.
Like amnesia.
Exactly. A skin cell forgets it's a skin cell.
Yeah.
It reverts all the way back to a raw stem cell and starts proliferating uncontrollably.
Oh, wow.
Yeah, this causes massive tissue hyperplasia and eventually highly aggressive tumors known as teratomas.
Yeah.
So temporal control, the exact timing of when the gene is on and when it is off, is literally a matter of life and death. You need to make the cell younger without making it a tumor.
It makes me think of the fountain of youth. If you find the fountain and decide to just drown yourself in it, you get cancer.
It's a dark way to look at it, but yes.
But this eye switch essentially gives the mice a little cup, allowing them to take perfectly timed microscopic sips from the fountain.
That's a beautiful way to put it. And the researchers figured out the exact rhythm for sipping. They subjected the mice to a strict cyclic EMF regimen. 3 days on end where the aging was reversed, followed by 4 days off, where the cells could rest and maintain their identity.
3 on 4 off. And what were the actual results of that schedule?
They were stunning. They tested this on 20-month-old naturally aged mice, which are essentially senior citizens in mouse years,
Right.
and on mice with a genetic mutation that causes premature aging, known as progeria. Under this cyclic regimen, the maximum lifespan of the mice was significantly extended.
Wow.
But it wasn't just about living longer. They lived healthier. The walls of their aortas thickened and regained elasticity.
Heart health improved.
Yes. The severe curvature of their aging spines was dramatically reduced. And crucially, a molecular marker called P16INK4A, which is a major red flag for cellular senescence and aging, absolutely plummeted.
And what about the tumors? Did they get cancer?
0 tumors over a full 6 months of testing.
That is wild.
It gets better. They even created a miniaturized, coin-sized EMF coil and placed it over severe skin wounds on the backs of older mice.
Like a targeted healing patch.
Exactly. By turning the switch on locally, they rapidly accelerated the wound healing, again, with absolutely no cancerous growths.
That is just stunning. So by perfectly timing a 3-day on, 4-day off cycle, they safely reversed physical aging. It makes you wonder, if you had the opportunity to spend exactly 3 days a week in a specialized magnetic field to safely dial back your biological clock, would you do it?
I think the line for that machine would wrap around the block.
No kidding. Now, logically, if we have the power to reverse physical aging, we can also use this tool to completely rethink how we study the diseases that are caused by aging. Which brings us to the second application, decoupling Alzheimer's disease.
Yeah, this is a monumental shift for neuroscience. Historically, modeling Alzheimer's in mice has been deeply flawed.
How so?
Well, the traditional way we study the disease is by genetically modifying a mouse so that it produces toxic amyloid beta plaques in its brain.
The classic Alzheimer's plaques.
Right, but because of how the genetics work, those mice start producing the toxic plaques from the very moment they are born.
Which is nothing like how human Alzheimer's works. We don't get these plaques in our 20s. We get them in our 70s and 80s, after our brains have already naturally aged for decades.
Precisely. The traditional mouse model forces a young, resilient, healthy brain to deal with the disease. And because of that, scientists have always struggled with a chicken and egg problem.
What's the problem?
Is the severe cognitive decline caused purely by the toxicity of the plaques themselves, or is it because an elderly brain has an intrinsically weakened, vulnerable microenvironment that just overreacts to the plaques?
I see. It's like planting a highly toxic weed.
Okay, go on.
If you plant it in fresh, nutrient-rich spring soil, the soil might be strong enough to fight it off or contain it. But if you plant that exact same toxic weed in tired, depleted winter soil, the whole ecosystem collapses.
That is a perfect analogy. And the ice switch finally let scientists test that winter soil theory.
How do they do it?
They attached the magnetic switch to a mutated human APP gene, the gene that generates the toxic plaques. But they kept the EMF turned completely off while the mice grew up.
So the mice were totally healthy.
They let the mice live totally normal, healthy lives until they reached 20 months of age.
The senior citizens again.
Right. Their brains grew old, their immune systems naturally weakened, and then in that already aged environment, they flipped the magnetic switch and flooded the old brains with the toxic plaques.
Wow. And what happened? How did the aged brain react compared to a young brain?
It was a violent overreaction. First, the aged mice produced a significantly worse ratio of amyloid beta proteins. They generated massive amounts of a beta-42, which is a highly sticky, extremely toxic variant compared to the less harmful a beta-40.
So just the chemistry was worse.
Much worse. But the real shock was the immune response. The aged brain suffered from catastrophic neuroinflammation. Microglia and astrocytes, which are the brain's immune and support cells, absolutely swarmed the plaques in a frantic, completely dysfunctional way.
Did that physical inflammation translate into actual cognitive decline for the mice? Could you see it in their behavior?
It did. They ran the mice through a Morse water maze, which is a classic test where mice have to swim and rely on spatial memory to find a hidden platform.
Right.
The aged mice with the newly activated plaques showed severe spatial memory deficits. They completely forgot where the platform was. This experiment definitively proved that the intrinsic vulnerability of an aging brain acts as a massive accelerator for Alzheimer's pathology.
That completely reframes how we look at dementia. Let's just pause again to make sure we're tracking the sheer scale of what we've covered so far. Time for our second quick recap.
Let's do it.
Bullet 1. We saw that a 3-day on, 4-day off magnetic cycle using the OSK factors can safely reverse cellular aging without causing cancer.
Very important.
Bullet 2. By waiting to flip the switch on the APP gene until a mouse is naturally old, scientists prove that an aged immune environment makes Alzheimer's exponentially more destructive.
Exactly right.
Knowing that a pre-existing aged environment accelerates the disease, it completely changes how we should think about long-term brain health and preventative maintenance.
It really highlights that preventing Alzheimer's isn't just about stopping the plaques. It's about keeping the brain's immune environment youthful.
Okay, so we've fixed physical aging and we've accurately modeled cognitive decline. But what about our emotion?
Mood is a whole different beast.
Right. If this magnetic remote control can change muscles and memory, can it actually alter mood?
It can, and this might actually be the most elegant application in the entire paper. The researchers wanted to see if they could cure severe genetically driven depression.
Okay, how did they approach that?
They focused on a gene called TPH2. This gene is the critical rate-limiting enzyme for synthesizing serotonin.
Serotonin, right, the classic happy chemical.
Exactly. As we know, serotonin is a primary neurotransmitter responsible for regulating mood, happiness, and anxiety. So they used a specific strain of mice that had a severe TPH2 mutation.
Meaning they couldn't make enough serotonin.
Right. These mice produced very little serotonin and exhibited intense depressive and anxiety-like behaviors. The team took the fast-acting version of the switch, the Psi system, linked it to a healthy TPH2 gene, and injected it directly into the dorsal Roffey nucleus.
The dorsal Roffey, what is that?
That is a structure deep in the brainstem that acts as the primary hub for serotonin production.
Wait, let me stop you right there, because my immediate thought is, this is simple math. The mouse is depressed because it doesn't have enough serotonin. We now have a remote control that makes serotonin.
I see where you're going with this.
Why not just turn the EMF on 24 hours a day, 7 days a week? Just permanently flood the brain with happiness, right? Cure the depression forever.
It is the most intuitive assumption to make, but, well, biology is rarely a simple math equation. And the researchers actually tested your theory. They left the EMF on continuously, 204/7, and it was a complete failure.
A failure? Why? What went wrong?
Because if you just leave the switch on, you cause extreme hyperactivity in the neurons. The brain gets completely overwhelmed. The continuous exposure didn't rescue the depressive behaviors at all.
Oh, wow.
You see, in a healthy, natural brain, serotonin levels are not a flat, constant line. They follow a strict circadian rhythm. They naturally rise and fall depending on the time of day, wakefulness, and environmental cues. By blasting the brain 24/7, they were completely ignoring the natural rhythm of life.
I see. It's like an orchestra.
An orchestra.
Yeah. Serotonin is the horn section. You can't just have the horns play at maximum volume for the entire 2-hour symphony.
Right, that would be awful.
That isn't music, that's just a blaring siren. The horns have to swell and fade with the rhythm of the conductor. Our biology demands that exact same rhythm.
That is a perfect analogy. You need the dynamic range. So the researchers adjusted the EMF schedule. Instead of 247, they programmed the magnetic field to cycle, 12 hours on end and 12 hours O off.
To match the day and night cycle.
Exactly. This perfectly mimicked the natural circadian biological rhythms of a healthy animal.
And did the orchestra play beautifully this time?
Flawlessly. Under the 12-hour cyclic regimen, the serotonin levels in the brain, along with its key metabolite, which is called 5-HIAA, were perfectly restored to normal physiological levels.
And what about the mice's behavior? Did they stop acting depressed?
The real proof was absolutely in the behavior. They put the mice through a tail suspension test, which measures how quickly a mouse gives up and becomes immobile. It's a classic sign of depression. The cyclic EMF dramatically reduced their immobility time. They also used a marble burying test, which measures obsessive, anxiety-driven digging.
Did they stop digging?
They stopped frantically burying the marbles. Their severe depression and anxiety essentially vanished.
That is just profound. By simply mimicking a natural 12-hour biological rhythm, this switch didn't just produce a chemical, it literally gave the animal its life back.
You really did.
It's wild to think that when a gene is active is just as critical as if it is active. Ask yourself, does it surprise you that timing is that important?
Timing is everything in biology. And the broader implication here is immense, because this EMF system showed absolutely zero adverse health effects or tumor generation over 6 straight months of testing.
6 months is a long time in a mouse's life.
It is. So the next logical steps are moving to large animal models and eventually human clinical trials. What we're looking at is a highly tunable, completely remote, bio-orthogonal platform for the future of medicine.
It is a complete paradigm shift. We've spent this entire deep dive looking at the sheer elegance of this gene switch. It entirely bypasses the sloppy side effects of chemical drugs.
Yes.
It reaches deep into the brain without the need to drill holes for fiber optics. And most importantly, it respects and operates on the precise rhythmic timing that our biology actually demands.
It works with the body, not against it.
Exactly. As we wrap up today, I want to leave you, the listener, with a thought to mull over. Throughout this journey, we've seen how a very specific 60 Hz, 2.0 millitesla magnetic field can act as a literal remote control for our DNA, tapping out a Morse code that our cells perfectly understand.
A secret language.
Right. We've always viewed the invisible forces in our environment as just the background noise of modern life. But if our microscopic cells are already perfectly built with antennas to decode specific invisible magnetic frequencies, what other hidden frequencies in our world are our bodies currently listening to? Thanks for joining us on The Deep Dive. We'll see you next time.